In this video, we will conclude this GlycoRNA topic. So, in this figure or in this video, we will discuss some of the additional approaches that has been used in the paper to confirm that GlycoRNA are present on the cell surface. And these approaches are this anti-RNA antibody, another one is Siglec binding. This is the anti-RNA antibody, or more specifically the antibody that is used is J2 antibody, and the function of this J2 antibody is that it will bind to the double-stranded site of the RNA, or the site of the RNA that is prone for double-stranded bonding, and also that RNA should be greater than 40 base pair. However, the RNA that is detected are the small RNA or the less than 40 base pair. But in one of our previous examples or previous figures, we have seen that there are some RNA that are detected that have high molecular that have high length, or we can say that have high base pairs. So this is the schematic overview of their experiment. They are expecting that RNA is bind to the cell surface. RNA is present on the cell surface that is bind to the glycan. So first approach that they use is they use this anti-double-stranded RNA to check whether there is a signal of this double-stranded RNA or not. And second is that they use this different Siglec. First specialty of Siglec is that it will bind to the sialic acid. So this panel B is shown the different combination of this anti-double-stranded RNA and RNase S on the flow cytometer. So for the unstained, there is no signal in the J2 site. However, when they add this RNase inhibitor, they can see some of the signals, that is 0.5%. This means that the reasoning for this they have mentioned is that these antibodies might contain some of the RNAs because of the crude preparation of process. So that RNAs might help in degrading these RNAs that are present on the cell surface. Because of this, there is no signal we can observe in the unstained one. So when they add this RNase inhibitor, we can see some of the signals, and when they include these antibodies along with this RNase inhibitor. So in this reason, the why they use this control is that I have mentioned you wrong description. The correct description is that during the sample preparation, the cells might be washed with PBS, and adherent cells might get removed from the cell surface with the help of trypsin. So trypsin might contain RNAs because of the crude sample preparation. So in order to block those RNAs, they use this RNase inhibitor. So from this we can see some of the RNase signal. And when we add this J2 antibody, we can see the RNase inhibitor, we can see there is increase in the amount of this J2 antibody signal. So this shows that GlycoRNA are present on the cell surface, and when they did not add this RNase inhibitor, there is a decrease in the signal. And when they add all the things such that is RNase inhibitor and RNase and J2 antibody, the signal is quite similar to the third and fifth are quite similar. So this shows, this panel B shows that the GlycoRNA are present on the cell surface as there is an increase in the J2 antibody signal. Next, they use, apart from these two, they also used another approach, that is from the previous findings of previous figures. They have experiments, they have identified that this GlycoRNA might process through this and glycosylation pathway. So they inhibited the OST, that is oligosaccharide transferase function, through this inhibitor NGI1, and then they check the J2 signals with, and they have found that with increase in the concentration of, and they have found out that with increase in the concentration of this inhibitor, there is a decrease in the signal. That we can see that when the concentration of this inhibitor, OST inhibitor, is low, the signals are high, and when the concentration of this inhibitor is high, the signals are low and it is going down. And these are the controls that they have used. So this panel C shows that the GlycoRNA or substantiate the point that they made in the previous figures, that GlycoRNA are processed through this asparagine linked glycosylation or N linked glycosylation. And this final panel, that is panel D, is based on the Siglec binding. So they use two different conditions. First is without any enzyme, and another one is with RNase. And if the signal with RNase reduces, then we can make a point that the Siglec bindings are without enzyme include both the sialic acid binding that are present on the cell surface, and also the RNAs with this glycan that contains sialic acid are present on the cell surface. So when they did the experiment with Siglec 7, there is no change in the signals, whereas for Siglec 14 we can see the change when they treat with RNase, the signals goes down. So this shows that when this signal, that is without any enzyme, might also be contributed by GlycoRNA. And same thing they have observed for Siglec 11. So from this figures, in overall they substantiate their point that GlycoRNA are present on the cell surface and they are processed via asparagine link glycosylation pathway. So if you need to read this whole paper in detail, you can go through the reference that has been provided in the description. It also includes some of the supplementary figures that I have not discussed in this GlycoRNA discussions. So that's all for this video. Thank you for your time.